Okay. Friends, actually, very good afternoon or good evening to all of you. I don't think actually we have anybody from the foreign participant today. So we have all from India. So I can say a very good evening to all of you. So we have today the 27th distinguished lecture being organized by INA Bhubaneswar chapter, SOA University, IIT Bhubaneswar, IMMT Bhubaneswar, and IEEE Bhubaneswar chapter. So this is actually 27th in the series of lecture that we had, and we have actually many eminent speakers here. And today we have got a very young and energetic speaker who will be talking to us on a completely different topic, something related to neuroscience and artificial intelligence and how these things could be utilized for the humanities. I think, and he is actually an authority on this to speak. So before we proceed, I will just briefly introduce Professor Tapan Kumar Gandhi, who is an associate professor in IIT Delhi, and he is working as an associate professor in Department of Electrical Engineering and cadence chair professor of artificial intelligence and automation, joint faculty at the School of AI, IIT Jodhpur, and sorry, at IIT, adjunct faculty in the School of AI and Data Science in IIT Jodhpur. So he is quite an accomplished person. He has published papers in top ranking journals like Nature, PNAS, Current Science, PLOS Biology, IEEE Transaction, etc. He has more than 200 publications in international journals and conference proceedings. Without actually going much detail into his long biodata, I would just like to mention a few things. He has 4 patents and 3 technologies that has been transferred to the Ministry of Social Justice, Government of India. He has received Platinum Jubilee Young Scientist Award 2015 by National Academy of Science, and is also an elected fellow of Indian National Academy of Engineering. Recently, he is the chairperson of the Project Prakash charitable trust that helps in restoration of vision and providing education to visually impaired children in India. A great job. So now I will like to say or invite our host institute, Vice Chancellor Professor P.K. Nanda, to say a few words. After that, today, our Director of IIT Bhubaneswar, Professor Dipat Kalmatkar, is also present. I request him to say something. I do not know actually whether Professor this Dr. Ramanuj Nair Narayan from IMMT Bhubaneswar, whether he is present. Otherwise, actually, we'll definitely ask him to speak something. Now, please go ahead, Professor P.K. Nanda.
Sir, thank you, sir. Good evening, esteemed chairperson, chairman of this INE chapter and chairman of our advisory board, Professor Damodar Acharya, esteemed director, Professor Sripath Karmalkar, and today's speaker, Dr. Tapan Kumar Gandhi. I know him well. He's a very prolific researcher. And Punda sir, namaskar. And all the distinguished INE fellows and participants. I think this is a very great opportunity to listen to many distinguished speakers on this platform. And SOE is proud to be part of this program. And SOE University also is quite proactive in conferences, seminars, and other things. And this year University is positioned 15th in the NR ranking. We are trying our hard to establish different research centers. We have 18 research centers, 50 research, 60 research labs, and different new centers also we are thinking of creating. And so that we have to have focused area of research. And the research area of Dr. Tapan Gandhi is very encouraging and very interdisciplinary in nature. I know, and very promising work he's doing, and direct societal benefit, like helping the visually impaired children in Delhi or in India itself. So I think university is quite proactive in taking up new research grants. So we'll be very happy to collaborate with Professor Tapan Gandhi. We have established a Center for Biomedical Engineering Application recently, and that some doctors and others also have joined there. So maybe in future we'll request you to help us go ahead in that direction. So thank you very much, sir. I have shared many times about our university, and he knows about our university. So thank you very much.
Okay. May I now request Professor Sripath Karmalkar, Director of IIT Bhubaneswar, to say a few words.
Professor Nanda has already spoken. The only thing I want to add is it is very nice to that here this kind of lecture series has been started, where we get exposed to current ideas, and that enriches us a lot. And I would like to see how I can contribute to increasing the audience for this. I think that is all. That would be, if I can do that, I think I would have contributed in some way. So I'm looking forward to hearing Tapan Gandhi. Last time, Ram Gopal Rao was there, and unfortunately, because I was doing faculty selection and that got extended, I could not attend. Otherwise, I would very much like to be part of this as in the audience. Thank you.
Okay. Thank you, Professor Karmakar. So, with now I can request our young and energetic professor from IIT Delhi, Tapan Kumar Gandhi, to present actually his lecture.
Thank you very much, sir, for this wonderful opportunity. And this is truly a not privilege; it's a blessing. I mean, to me, because coming from Odisha and finally recognized by this the best society, INA Society Odisha chapter. It's a very big privilege for me. And thank you very much to all of you, to all of my teachers, and the eminent scientists of Odisha, and also from other part of India. Those who are going to listen to my lecture, and please stop me if I am not able to convey the right message from my talk. So let me share my screen and then I'll start. Sir, I hope you can see my screen now. Yes. Okay, sir, I can start. So the title of my talk is Advancing Science and Serving Humanity. So here I would like to start.
Excuse me, can you go full screen if it is possible?
Sir, this is full screen.
Okay, thank you.
You can make it full screen.
This is, I have already made it full screen.
But
okay, thank you. It's all right.
Not coming?
No, it's
no,
okay.
we are not getting full screen.
Okay, I'm
Sir,
now.
I am able to see full screen. Yeah.
Okay. Uh, so the title of my talk is Advancing Science and Serving Humanity. Here I'll start the day I decided to be a scientist and how I came, I mean, through a long way in this domain. So when I was a student 18 years ago, one of the fascinating problem for me was to work on the spinal cord injury patient. So first time when I came to IIT Delhi during my MTech time, I was exposed to the different problems in the hospital, and the problem which which really touched to my heart is the spinal cord injury related problem. Because that time I met many people, those who are having the spinal cord injury. So this is the spine you can see in this picture, and these are all different vertebra, and all these are connected to different parts of our body. So if any injury will happen, any part of this vertebra or the whole vertebra, the person will be paralyzed, maybe partially or maybe fully paralyzed. So why this problem was very interesting to me, because that time I thought, let's say if somebody born all right and spent maybe half of his life as a normal person, and because of this problem, the person is not able to do anything or not able to speak or not able to do the daily life, daily things, then it's really very difficult for anybody. So being an engineer, I thought that is it possible to develop some kind of technology by which we can help them to do their daily things. So that time I developed three gadgets. So first what I did, I observed that what are the residual ability present in this patient. That means those who are spinal cord injury, um, uh, maybe they are not able to walk or maybe they are not able to speak. So I try to understand which is the residual ability present in those patients, and using this residual ability, can I develop some gadgets? So that time in 1 year, I developed three gadgets. One is I call the blow switch. That means just giving a blow. Let's say if you are alone at home and you want to switch on any devices or you want to control any gadgets by blow, you can control the gadget. So you can see here the website. Actually, these are the pictures from the hospital from 2004-5. I did this trial. Then another device was called the touchpad. Touchpad means, so that time one boy from Bihar, he was also spinal cord injury patient, and then when I met him, he told that my family, when we were traveling on road, we met an accident, and I lost my ability to control my hand. So I love playing computer games, but now I cannot control the computer key or I cannot do any computer job. So that time, inspired from this kind of problem, I developed another gadget called the touchpad. So just having this 5 touch, you can see here, this was my first prototype. And using this, those who have no, I mean, those who cannot move their fingers and not able to type properly, they can do all their computer jobs. They can surf the internet, they can write everything just using this 5 touch keyboard. So that time also, I met some soldiers who fought the Kargil war, and they were being treated in the Apollo Hospital in the Spinal Cord Injury Center. So there, what I found, the person, they're completely paralyzed, but still they can move their eye. So I thought, can I make some control signal or can I generate some control signal from the basic eye movement? So you can see here in this picture, when we rotate our eye left to right in a normal movement, it generates some potential, and the potential is very tiny. So the question is, can we amplify it or can we generate many potential or many signals out of this basic few eye movements? So using some automata, I mean techniques and some behavioral technology, that time I generate more than 100 control signal from the basic eye movement. So that time when I developed all this gadget, actually this was just using this eye movement, somebody can control the wheelchair, and if somebody is not able to speak, they can display the message that what they want. So everything they can do just by eye movement. So during the time when I used to go to the hospital to test on the patient, so being the inventor, I knew what are the lacuna of my devices, because these are all learning based. You have to learn, then only you can use it in a better way. So that time one question came to my mind, that is it possible that instead of learning this device, because you know that this learning ability is not equal to all patients. Some of them learning very fast, some of them learn very slow. So that time one thing came to my mind, that is it possible that I can make a device which can understand the intention of the patient and accordingly it will behave. So like this, the patient is on the wheelchair, and let's say this is a device in front of him or her, and the device will understand what's the need of this patient. Just like, let me give one simple example. Let's say sometimes when we are ill, and let's say I'm in my study room and my mother will come with some drink or maybe some juice and tell, oh, this is, then automatically it comes to our mind that how my mother knew that I wanted some kind of drink, right? So likewise, this is the device will understand what is the need of the person and accordingly it will behave. So when I was doing this kind of research that how I can make this kind of device, I found then the answer, obvious answer was the artificial intelligence. And when I interacted with many eminent professors in this domain, and then when I tried to enter to the domain of artificial intelligence, I found nothing in the domain of artificial intelligence. Rather, when I enter more and more, I found that there is only one way to advance this domain of engineering. I need to understand the real intelligence, and this is called the brain. And that's why I've written here inferences from brain science. That can we understand the true intelligence, and then maybe if we'll understand the true intelligence, we can mimic in the machine, and this is how we can create the real kind of intelligence. So fortunately, I got an opportunity to work to do my PhD with MIT, and that was on the brain. So the brain is a very small structure. This is just 2% weight of our whole body, but it consumes nearly 28% of whole energy what we needed for our everyday tasks. So that means brain is doing many remarkable job which we need to understand, and by that we can advance the engineering. In the same time, we can answer many interesting questions of the neuroscience or the medical science. So here in my lab, I focus on three main important kind of work. The first one is to understand how the brain develops. That means after the birth of a child, how the brain is developing and how the brain is learning. Second is to understand normal brain functioning. Let's say being an adult, if we believe our brain is normal, then how it functions. And third is to understand brain disorder. If the brain is having some kind of problems, then how it behaves. So if we understand these three different kinds of brain, most probably we can take lead both in health as well as in engineering. So why I'm talking about health and engineering? Because in all the out of all the healthcare problems, the most devastating one is the neural related problems. Any disorder or any problem related to neuron is the problematic. There is no solution, direct solution for those problems. And if you look at the engineering, the most sophisticated domains are AI, computer vision, robotics, which I mean everybody is coining nowadays. So in both this, whether health or engineering, one common thing will come. This is called the intelligence or the neuron. So that means we need to understand the true intelligence of the neurons, how they work together, and in that way we can truly contribute in the advancement of fundamental science in the domain of healthcare as well as in engineering. So here I'll bring a very interesting work I do. So I thought that this would be more appropriate to highlight, because as a scientist, always we do research, and many times also we have noticed that many of our research does not get a chance to see the daylight. Uh, Even many of our research has no end goal. But this is the example I'm going to give you, all of you here, that how doing humanitarian research we can contribute to the fundamental science. In the same time, it helps us to develop the next generation technology. So here I'm in this picture I'm showing you. This is very, I mean, one kind of not a good scene about India. So one in every 100 Indians are blind. So you'll be surprised to know that nearly half of the blind population of the whole world, they lives in India. And the causes of this blindness are called the corneal scarring. That means if there is any injury in the cornea, cataract. All of us know about old age cataract. That means always in the newspaper comes, or I mean, also people when they get older, they get the cataract. But actually, the cataracts also happens since birth. So many child take birth with cataract. Then because I mean this cataract develops for many reasons, because also you know that in India many marriages happens consanguineous. That means marriages happens within the I mean same religion or same relatives. That has a great chance for this cataract kind of blindness. Second is, you know that 80% people lives in villages and where the hospital are not properly sanitized due to the infections also. There's a high chance of getting the cataract when the child in their mother's home. Third is another is congenital rubella syndrome. The mothers are not vaccinated. That means when they are pregnant, they need to get vaccinated, and because of this lack of awareness, they not get vaccinated. And all of this problem, India has a huge amount of childhood blindness. So that's why, because of this childhood blindness, so these are the consequences. You can see here, the lifespan of the blind children, those who born blind, their lifespan is 15 years less compared to the normal counterpart. The childhood mortality is also very high. That means 50% of the blind children born, they don't get a chance to see their next birthday. Third is, because they're blind, their education prospect is also very less. And that's why the employment perspective is also very, very less. And because of this blindness problem, India is losing more than 20 billion US dollar every year. That means it's a very huge loss for the country like India. So that's why the big question is, pressing humanitarian need is to provide the treatment to this curably blind. If it's possible to cure this blind, so we should provide them the treatment. But this was not possible until we started our work based on this important hypothesis given by two Nobel laureates. So here in my slide, hope you can see it. So I'm showing two Nobel laureates, David Hubel and Torsten Wiesel. They got Nobel Prize in 1981, and they talked about one very important hypothesis called the critical period. So critical period hypothesis is considered as one of the biggest hypotheses in the domain of biology or medical science. So it says, let me give in my own, in my layman language. So let's say today I took birth, and after my birth, you put me in a dark room, and in the dark room, you provided me food, drink, whatever I need for my survival. And let's say I spent 2 months in the dark room. I was not allowed to see anything. So after 2 months, if you'll bring me to the lighted part of this world, by that time, I'll lose all ability to see. That means our sensory modality, all this, whether this is vision or audition, tactile, all this develops in the past few days or past few months of our life. So if somebody is not being or somebody is deprived of any of this information, that means they are not able to get those information later part of the life. That's why when somebody born blind, when they comes to the hospital at the age of let's say 5 years or 10 years, the doctor used to say it's not possible to restore sight to them. So all this research comes from all these articles you can see here. These are all very big journal publication. But one thing which we found very interesting, that all these results are from the animals, and all these results were extrapolated to the humans. So that was only one clue to us that what happens to the human, that if humans are born blind, that whether they are also following the similar trajectory of neural plasticity. And this work we started to understand more about the vision science and understand more about the vision. That being a human, let's say, if I'll ask you one question in the domain of engineering, that how we learn to see. That means how we learn to see this universe. This is a very big question in the science, and we found it was not addressed properly, or no scientist has attempted to look at this problem because of many reasons. First thing is, if we want to look at this problem, that how we learn to see, that means you have to do research on a newly born baby. But it is, one kind of impossible to do research on newly born baby for two reasons. First thing is, when people do some research on a newly born baby, they will not understand what we are going to do. That means we will not get the reliable data. Second is, even if you will get some reliable data, but the ethical committee will not allow you to do research on a newly born baby. And that's the two most important reasons that why no substantial data is there to understand how our brain learns to see. Because when we talk about the artificial intelligence, before that, the natural intelligence coming. That means in the natural domain, all the sensory information, how we learn to hear, or how we learn to see, or how we start learning the feel, or the sense of smell, or the olfaction, all these are very, very big questions. So these are not addressed properly. So in this work, we thought that we should take a challenge. And when we started this work, that time we are criticized by the whole world that how come you can work against this Nobel Prize winning hypothesis. But we convinced ourselves that yes, there is some possibility that we might see some light out of this research. And then finally, we started doing this work. It's a humanitarian scientific project. Actually, this was started by Professor Pawan Sinha, who is my advisor, PhD advisor at MIT. Then I joined with him for my PhD, and we started working on this blind children. So the project name is called Project Prakash. This is headquartered in Delhi, and the meaning of Prakash is to bring the light in the lives of the blind. That's why we call Project Prakash. This now we have been big trust, non-profit trust here in Delhi, and I'm heading the trust. So in this trust, what we do outreach. We have a very big team. they go to different places in India, different state, like all the adjacent state of, near Delhi, like Rajasthan, UP, Haryana, Madhya Pradesh, and then we do screening of all these children, and then we bring them to Delhi for the treatment, and then do research, and finally we provide them education because they're all blind, they've not been to school. So when we started this work, that time we were thinking that we will answer the most scientific, interesting questions. But later on, we realized that when somebody has not been to school, after getting back the site, what they will do. Then we thought that we need to integrate the education into it. And just doing all this thing, we are getting many opportunity to address many important questions of the science. And maybe I'll highlight some of them in my lecture. This is the treatment part. We just provide some kind of basic treatment, and then we try to understand how, whether the person can learn and whether the brain is able to reorganize again to see this beautiful world or not. So that's why the first fundamental question here I'm asking here, that whether the vision develops after sight onset. If not, why? And if yes, how the vision is developing. So we have, until now, we have restored sight to more than 700 blinds in India. Those who are born blind, and they can see just like you and me. And we have provided many surgery, provided many non-surgical treatment to many patients. And we are, every year, at least we are restoring sight to minimum 20 blind subjects. And depending on the funding, how much fund we raise, based on that, we do the treatment. Because for each patient, we have to spend huge amount of money for their treatment, for their all expenses. That's why we cannot go beyond more than 20 or 25. We do many different experiments. We image their brain, and we also collect the brain signal. Also, we do many computational experiments to understand better that how the brain is learning. So this is one of the study I'm just highlighting here. So I'll not talk much, but this is the images. You know, this MRI, this is a medical imaging facility where we image the brain. It helps us to get thousands of images of the brain, and then later we do the processing, and then we find out the neural connections, that how the neuron, this activation evolve. So for the simplicity, in this picture, I'm just showing you this what called the resting state functional connectivity. This is one of the most, very important approach, or I can say very strong kind of evidence through which we can tell many things about the brain. And interestingly, this hypothesis, or this, interesting aspect of the brain was first given by one scientist from Odisha. His name is Professor Bharat Biswal. So he's professor in US, but his technique, initially it was not accepted by the Western world, but now this is one of the most important techniques in the brain imaging. So using this technique, you can tell many things about the brain. So what it says, this hotspot, you can see some color picture. This is the brain in two different sides. The first picture is, if you look at the brain from the left side, and the other picture is from the inside. So here, the red color or the yellow color, what you see here, that means though they are located in different parts of the brain, but they are functionally doing the similar job. So if I'll take a time series signal from the yellow color region, let's say PCC here I've written, or MPF, they are highly correlated, you can see in the signal. But if I'll take a signal from IPS, let's say another region which blue, this is anti-correlated. That means depending on the correlation, we can create the functional network of the brain, that how the brain does all remarkable job. So we use this simple technique in some analysis. So this is a very interesting picture I'm showing you here. Why this is interesting? Because around 40-50 years back, the great grandson of Charles Darwin, his name is Horst Barlow. Horst Barlow is the professor at now emeritus professor at Cambridge University. He came up with one hypothesis to understand that how brain encode the information. And until we answered this question, it was not known to the whole world that how the information encoding happens in the brain. So what I found here, so this is what I'm showing you. This is a 21-year-old. That means a person who is 21 years old, being born blind and blind until 21 years. We brought him to his place. He's from Rajasthan to Delhi, and then before the sight onset, before restoring the sight, what we did. So T-2 means 2 days before surgery. That means the person is completely blind at the T-2. T plus 7 means at T equal to 0, we restore the sight. And after the restoration of the sight, this is T plus 7, 7 days after the surgery. T plus 30, 30 days after the surgery. And T plus 120 is 120 days after surgery. So what this brain picture says, this is the back part of the brain, which is responsible for the vision. So you can see here some hotspot, the yellow, red, and some cool color. So the hotspot, this you can see this big hotspot T minus 2, and then again after 7 days of the surgery, the hotspot again becoming a little bit larger, and after 30 days you can see the hotspots are getting fragmented. You can see this is getting, I mean, you can see more fragment, and after 120 days of the sight onset, you can see many small fragment kind of structure. So what it says. So I'm just giving one very small example from the electronics, how we encode the information. Let's say if we have two bits of information, that means we can encode in 4 different ways, depending on whether the bits are dependent on each other or whether the bits are
I'll call him back.
Am I audible?
Yes, yes.
Okay. So if I have 2 bits, if the bits are dependent on each other, then that means they can encode only 2 information, either 00 or 11. But if the bits are independent, then they can encode the information in 2 to the n, just like 00, 011, 011. So what we found that when the bits are independent, they can encode more information. So this is exactly what is happening in the brain. So when the person is blind, that time the neurons are actually they are dependent on each other. That means they are only two big blobs. You can see here 2 big blobs. After 7 days also there is not much changes, but after 30 days they're becoming, I mean, out of this 2 big island, we are getting a little bit more island, and after 120 days you can see that there are no big blobs of hotspot, only the small fragmentation. That means slowly the neurons are getting more independent. That means the neuron are encoding more information. So we found from all the subjects, here I'm just showing you, this is the graph I'm showing different color of the different subjects to whom we restore sight, and the black color is the mean of all. So what we found along that post surgery, that means when we restore sight and we follow along the longitudinal timeline, and what we found that in the brain there is a decorrelation. So more decorrelation means more information encoding in the brain. So this is the first kind of, I mean, we provided the scientific evidence that how the brain is learning. So, and this was also scientifically also good way to tell that the brain is plastic, or the brain has the ability to change even the person has not seen until the age of 20 years. So we have published, actually this is not only the one research, we have done many different research and published in many top-notch journals like Nature, Science, and Current Biology, PNS. So from all this kind of results, what we found that the brain is very plastic enough to make use of information from the age later on life. That means whether it's 8 years or 22 years, the age is not the big things for the sight processing. We found even up to 25 years, we are able to restore sight to the person who has not seen anything. That means the neurons are very plastic in nature. So later part of the life, they have the ability to see again. Then here, let me change this slide. So actually, I want to give one example that how we work at the interface of fundamental neuroscience and artificial intelligence. So let me just change this slide, and then I'll come later this slide. I'll give you another good example here. Just 1 second, sir. So here, I talk about a very interesting problem in the domain of both computer vision as well as in the neuroscience. This is called the color perception. That how we perceive the color. So this is a very interesting study. Recently, we submitted to Science this work. That's why I thought that it would be interesting to share with all of you. Okay, so here I'm showing you that how we learn the color perception. So here, what you see in my screen, this the newborn baby. So when a baby take birth, so that time their vision is completely blurred, and that time they cannot see any color things. So you can see here in this picture, the first picture is newborn, then after 4 weeks of the vision, then 8 weeks of the development, 3 weeks, 6 months, and adult life. So you can see here how the chromatic information are getting added to their perception. So when we are adult kind of adult stage, then only we have a good chromatic information. So this is how the vision, develops, the color perception develops in the human being. So if I look at the eye, so this is the eye. I'm giving some little bit of background of biology because I know that all of us are engineers. So here you can see this is the structure of the eye, and this is the front part, and this is the back part. Back part is the retina part. You can see. So the rays of light enters to our eye and falls on the retina, and then retina converts this light energy to neural energy, and it passes this neural energy through this optic disc. You can see here through optic nerve to the brain, and all these beautiful things happen. So why this is very important? Because you just see, when you see some object, the rays of light enter to our eye, and something happened in eye and brain, then we see what the object is. So just imagine what we see, maybe it does not exist, or which exists in the world, maybe we are not able to see it. That means if our brain and eye will decide what to show us, then only we'll see. If they will decide not to show anything to us, then the whole world would be completely dark to us. So that's why it's very, very important to know that how this range of light is being perceived as different objects, what we see around us. So this is a very, very fundamental question and interesting question in the domain of science. So coming to this color perception, as all of you know, that we see color because of the two kinds of cells present in our eye. One is called the rods, another is called the cone. So you can see here, I'm showing you the newly born baby. When the baby took birth, so their cones are very fat. You can see I'm just moving my cursor. And when we become adult, let's say if I look at your cone or my cone, our cone is very thin and very elongated. So when it's fed, so the packaging is not very tight. You can see in the right side two picture. If you look at the lattice, this is how the cone cells are packed. That means it is not packed tightly. But when through our process of this development, when we become the adult, like you can see, this is a very small and very, very tightly packed. So what happens when the child takes birth? Only they start learning this color perception through the grayscale images, because that time also the cone lattice are not matured, and through this maturation, same time the color component get added, and finally we get the complete vision through which you can see the true color, right? So this is how it's developed. So what we found, so this is actually what I'm showing you, two different images. One is in the color and the same images in the grayscale. So when you treated these blind children, those who are born blind and to whom we felt that we can restore sight to them, when they start seeing this world, we presented these two kind of images, color and grayscale. Interestingly, they can recognize well all the color images, but they fail in many grayscale images. So that make us to think that why they are failing in the grayscale images. So for that, what we did, just like here, I'm just showing you some picture database. Actually, we use this data set both for the human as well as in the domain of AI or the machine. So we took these images from the ImageNet database, and then we tested on both of our subjects. I have written here Prakash subject. Prakash means to whom we restore sight. Our project name is Prakash. That's why I have written Prakash participants, and here is control. So for all these images, there is no difference in gray or color in the control when we tested. But what we found with the Prakash participant, that means those who restore the sight, we found they're doing very good in color images, but they are failing in the grayscale image. So here, this is the question that why they are failing in the grayscale image. So here, coming to this picture. So as I told you, that newly born baby start having very big cone structure, fat cone structure, and adult is very thin. And when the baby take birth, that time their perception is like the left side picture, you can see. And through the aging process, they develop this color perception, and finally they see these color images. But when we restore the sight to a 20-year-old person, let's say 20-year-old adult, that time his cone maturation has already happened, but he was deprived of any functional vision. So when we allow them to see the world at the age of 20 years, they first get exposed to these color images, the last category of images, right? So that's why they're very good in color. So what we did, to understand more about this learning process, that how the human learn and whether the similar kind of learning is happening in the machine or not, we use simply the ResNet. I'm not going into details of the machine learning. We took this standard ResNet network, and then we trained this network with the different sets of images. So here, these are the images we took. So here, I've written C to C. C to C means color to color. So I have divided my training paradigm in two halves, first half and second half. So in the first half of my training, I trained my network with color image. And again, the second half, I trained with color image. Then G to G, color to gray. No, sorry, gray to gray. That means first half gray image, as well as I trained the second half also gray image. Then what I did in third case, I first trained with grayscale image. Then second half, I trained with color image. Actually, this third category, G2C, is the true representation of how, as a normal human being, we learn the color. So G2C, this is the third category. And then color to gray. That means first half we trained with color images, and then second half we trained with the grayscale images. This is how this our Prakash subject. That means those who start with, those who are born blind and started their learning after 20 years old age. And what we found, this is a simple picture I'm just showing you here. So when 0 is hue, this is the hue rotation. So when you can see here, in this case, when there is no hue rotation, in all these cases, they are performing well, the model performs well. But you can see here, when we are trying to rotate the hue, then the accuracy of performance is decreasing. In case of gray, color to gray, you can see here, and color to color. That means if the network is trained with color images first and then color images, the performance is not robust, or the model is not robust, as well as if the model is trained with color, then gray. But in the other two cases, if we are training our model with grayscale images in both the phases, then it is doing well, as well as if we are giving the first half is grayscale and second half with color images, then they are also doing well. So that means whatever the inferences we are getting from the human, from the biology, also similarly applies to the machine learning model. So generally in the artificial intelligence, the machine learning, what we do, many times we do randomly, that how it works. But actually, if we understand the fundamental of this intelligence, that how the natural intelligence work, then most probably it would be much more robust, just like one example I gave you. So likewise, we have developed many other models where you can take the inferences from the biology and you can apply to the domain of AI. So then again, I'll go back to my the previous slide. Okay. So I hope that you got the idea that how we are getting the inferences from the biology or the science from this brain science and applying in the domain of machine. So yeah. So then I'll just highlight few other things. Okay. So here I'll just give you another very interesting example that how we can model an autonomous engineering system which are inspired by human external data. So one of the biggest question in the domain of AI is that how we can integrate all the sensory information to the machine. We humans are very intelligent because of the remarkable ability we have, just like how we exchange the different sensory modality. Just like if I'll give one example, let's say when I'm talking to our mother, let's say my mother is 100,000 miles from here. And when I talk to my mother, when I hear her voice, I can visualize her. So that means I'm doing the cross-modal mapping from audition to vision. Likewise, let's say if I'll ask you, close your eyes, and I'll give a mobile phone on your hand, and I'll ask you, can you please tell me, or can you visualize what I've given on your hand? And you can easily do it. This is called touch to vision mapping. So this kind of mapping is very interesting in the human. And until now, we don't understand how this is mapping is happening. And if we can understand, most probably we can take our AI to the next level. So here I'm giving one very interesting example here. So here you can see the child is holding the mobile phone. So here the big question is, how do we predict the visual appearance of an object based on how it feels to the touch? Is this mapping innate or does it need to be learned? So this is the crux of the AI. So that means whether we need to program our model in the past day to do all the intelligent thing, or we need to program a little bit and then we should force our machine to learn by its own. And this debate is a very, old debate, and still it's happening, that which are the ability we learn by birth and which are the ability we learn through experience. So here I'm just giving very interesting example. You can see here when the child is holding the mobile phone, and if we replace the finger by some sensor, we are getting some kind of haptic features like H0 to HN. In the same time, the child is also looking at the mobile phone. That means we are getting some visual features, 0, V1 to Vn. So what is happening here? Somehow, from this time series signal, the brain is able to create a map from haptic to visual features. And that's how the child is able to feel whatever he is touching. So this is a very interesting question. And this kind of question was posed by a very eminent scientist known as Molyneux. Molyneux in his letter to John Locke. So John Locke is another scientist. So Molyneux wrote a letter to John Locke in 1688. That means nearly 300 years back. Molyneux wrote a letter to John Locke having the similar kind of problem. And the problem is written over here. You can see here. And I'm telling you in my own word. So just imagine, let's say I'm born blind and I have not seen anything. Since birth, I'm blind. And today I'm sitting in front of you and you gave me one sphere and one cube. And you told me, just touch these two objects and feel and try to remember this is a cube and this is a sphere. So today I'm blind, I cannot see. So by touch, I remember that what is cube and what is sphere. And just imagine tomorrow, all of a sudden, I get back my light, I get back my vision. And if I get my vision and if you place these two objects in front of me, and you will ask me that don't touch these two objects, look at these two objects and tell us which one is what. And here the question is whether I'll be able to tell just looking at these two objects that this is a cube and this is a sphere, or I cannot tell. So this is one of the very interesting questions in the domain of the perception. And that's why it's already in the Stanford Encyclopedia of Philosophy. It's mentioned that there is no problem in the history of the philosophy of perception that has provoked more thought than the problem that Molyneux raised in 1688. So this is one of the very interesting the kind of question asked by some scientists 300 years back. And since the time, many people attempted to answer this question, and also they published paper in Nature and Science kind of journal. But the answer was not still debatable. No end to this question. Finally, working with these blind children, we did some experiment, very simple experiment. And what we found, so here what I'm showing you in this picture, I'm not going details into the experimental paradigm. What we found, you can see here, this short form YS, BG, these are the subjects' name. And we tested on 10 subjects to whom we restored sight. We experimented this, and what we found, that this subject, after the sight onset, they are not able to tell which one is what. But interestingly, within 7 days, they learn this ability. So what we found, that to the question which Molyneux asked, we found that the Molyneux question has a negative answer. That means that the person, after getting with the sight, they are not able to tell. But this neural learning is happening very quickly. Within 7 days, they can tell that which one is what. So and finally, we published this paper in Nature Neuroscience in 2011, and this opened up a very big debate that how come this neuron learns so quickly. And the day we published this research, it got highlighted in the front page, the whole front page of New York Times was given to our work. And it was mentioned that finally, after 300 years, the blind children of India, they answer this question, big question of the science. But it has huge connection with the domain of artificial intelligence. I'll come to that point after 2-3 slides. So here I'm presenting two pictures. The two picture you can see, and there are two names, Maluma and Takiti. So if I'll ask you all that, can you please tell me which one is Maluma and which one is Takiti? So this is a very interesting problem, and many researchers in the domain of computer science, they've studied that from the name how we can tag that which one is Maluma and which one is Takiti. So from the research, people found let's say just like I'm giving you two name and two object, and you have to tell which one is Maluma and which one is Takiti. Maybe you must be thinking. So most of the people told that the left side is Takiti, the right side is Maluma. Because it comes to our mind because it looks like Maluma is smoother, Takiti is for a zigzag. That's why Takiti is the left side. And this is most of the people told. So here the big question is how our brain thinks that this is Maluma and Takiti. Maybe it taking the inferences from our previous experience or maybe our understanding. That's why we are taking the name, the left side is Takiti and the right side is Maluma. So this is what I was posing just few minutes back, that how is the haptic to visual mapping acquired after gaining sight. So just, I'm just giving another example so that you can understand better. So every day we watch television. So actually this is a study I conducted around 15 years back. Today I'm just presenting. It's very interesting. So when we watch the television, let's say one news reader is reading the news, and always we feel that the voice comes from the mouth of this person. But if you look at the television, the voice always comes from the speaker, which is placed at the side of the television or below the television. But we feel that sound is coming from the mouth of this person. So this was a very interesting question at that time, and I thought that why we feel that the voice is coming from the mouth of this person. So then what I did, I divided the whole visual space into different, let's say this is a matrix, let's say 1000 by 1000 matrix. And I thought, if I'll observe each and every pixel of this 1000 by 1000 matrix, if I'll observe this each pixel, so I'll get a time series signal. And the same time, I recorded the voice signal. And then what I did, the top one, actually the first picture is the voice signal. I took the envelope of the voice signal here, the red color. And let's say if I'll take one pixel, let's say the yellow color is my one pixel, right? So I'm getting one signal like this. So likewise, let's say if I'll consider 3 pixel, I'll get 3 time domain signal, and the top one is the voice signal. So likewise, from the whole visual space, I can get, let's say if I'm dividing my visual space into 1000 by 1000 point, then each point I can take as a pixel so that I can get this blue color signal. And if I'll do the simple correlation between the red color and the blue color, I can create the picture like this. And this is a very simple technique we use in the computer science. So here, the hot color represents the higher correlation and the cool color represents the low correlation. So from here you can see we are getting the higher correlation around the mouth. That's why we believe that the voice comes from the mouth. Actually, this is the work I did in 15 years back, but now actually today I can see the application, big application, just like the OTV in Odisha, they first launched that how the AI Lisa can read the news. So this is one kind of inferences from this kind of study. So here, here actually the letter is not visible by somehow. So here what is actually from this kind of experiment, you can create a lookup table. Let's say for the mouth configuration X, what is the sound configuration? Let's say if I'll make a lookup table for mouth configuration this, my sound would be like this. For mouth configuration of this, sound would be like that. So if I can create a lookup table of, let's say, n number, so at the end, if I'll ask for this kind of mouth configuration, what would be the sound? I can easily correlate and I can easily tell. So from all this experiment, what we noticed that if I ask this question that how this cross-modal mapping established, I found the dynamics plays, the time domain dynamics plays a very important role to find out the correlation among all this kind of signal, and finally we can execute the task. So like I just talked about the Molyneux query, what the Molyneux asked, that whether the person which they know by touch can see, when they see they can tell or not, we found that no. That means even the person is born blind and being blind for 20 years, but he knows all this modality by touch, but still he was not able to map from vision to tactile or audition just after the sight onset. That means we should not give the all knowledge to the machine at the first step. So maybe, from all this experiment what I found, we need to give some kind of information or some kind of knowledge, very basic knowledge to the machine, and we should force the machine to learn by its own through this time domain dynamics. And in that way, we can make it intelligent. So here just I throw some light on this problem, but we have done many research on many studies, and all the studies are very remarkable in terms of contributing very fundamental science to the domain of medical science or the biology. And at the same time, it is helping us to push the AI to a little bit higher domain. So this is what I'm talking about. This is the work got published in the front page of the New York Times when we solved this problem. As well as the Science magazine cover page in 2015 was given to our work on the blindness. As well as our research has already been published in Nature Times magazine, as well as Harvard and MIT News. This is one picture from my lab at IIT Delhi. One German magazine, Geo magazine, came to cover some of the story that how we are trying to understand the natural intelligence and how we are advancing the domain of AI. This is one of the snapshot of that magazine. And then finally, I would like to say one thing which Albert Einstein says: those who have the privilege to know have the duty to act. So there's the same boy who came to us, born blind, and after the sight onset, you can see the smile in his face. So this is the trust I was talking about. Through this trust, not only we restore the sight, also we bring the people to whom we restore sight, we provide them education, so that we believe that we can bring them the real mainstream society. So in this way, we can have the broader impact. That's why I always believe that through illuminating lives, we can illuminate science. And in that way, not only we can advance the science and engineering, but also truly we can contribute to our society in a very big way. Thank you very much for your time and listening to me. And I would be happy to answer any questions if you have.
Thank you, Dr. Gandhi, for such a illuminating lecture. Definitely, it is absolutely like Greek and Latin for a Athoda engineer like me. So, but it is very, very interesting. Now, actually, I will like to say participants who are there and who are from this domain now ask some questions. Yes, Sanat Kumarji, please. You are mute.
Yeah, okay, now?
Yes, yes.
Yes,
Okay.
sir, I can hear you.
When you're talking about a newborn baby,
Yes, sir.
trying to understand things, I don't know whether you have noticed small children. This is not exactly a criticism or something. I'm just adding to your thing.
Yes, sir.
A small children, when you give something, they first put it in their mouth, and then they taste it, and then only they will understand what it is. Anything you give, the first thing that goes is it into that mouth. So there is something seems to be taste is also you said tactile, but taste is also seems to be feedback for the vision. This is point number one. I needed another point to be said. You are talking about a sphere and a cube. Okay. Now the blind person who is seeing it for the first time, he has to be told what is the definition of a sphere, what is the definition of a cube. So a cube is not a smooth surface. You at least you should tell him the definition, otherwise you will not know. Your other Maulan, what is that one? Tic tac or something like that you said, no? So there, what you said, I agree. Corners, I also thought of it that only. So why you said that? But for a blind fellow who is seeing something for the present, he should know what do you mean by sphere. So he should know a sphere is smooth or it doesn't have edges, something like that. Edge is there or not, he will find only by tactile, and tactile will not help him. So you have to give him a define. I don't know whether you have done this. Thank you.
Thank you, sir, for these two important questions. Sir, actually, you are right that the baby, when they, the baby, they put everything in their mouth. Though there is no strong evidence, there are many psychological study on this. There is no strong evidence, but from my understanding of this neuroscience, what I believe that when we started, actually, if you look at the single cellular organism, they also start relying on the chemical sensor. Chemical sensor, matlab taste or olfaction. And these are the first two sensors, the primitive sensor. But if you look at this multicellular organism like us, human, for us, the most important sensor is the vision. So maybe that baby, they are getting some inferences from the when they're putting their mouth. This is working well. Of course, I'm completely agree with you. But yes, I mean, more research is needed to understand, because this is one of the hottest area nowadays to understand the sensory mechanism. And coming to the second point, what we told, always when we give this sphere or the cube, we always explain what does it mean. But the question was that when they understood the concept, just listening and touching it, and let's say second day when they started seeing this world, we just allow them to see it without touching. We told them don't touch because they know by touch, but they could not connect. That what, because if we allow them to touch, they can easily tell. But just looking at the object, they cannot tell. So this is what we are calling.
By looking at it, can't they say this has got edge and this doesn't have edge? A cube has got edges. A sphere doesn't have edges. So can they not tell by just
all
looking
these
at it?
features, but sir, all these features they are able to see. Even the cube and the sphere, all the features, whatever there, what they've touched, they are able to see. But the what they see, they are not able to map to the tactile domain.
Okay.
I hope you understand. Yeah.
Understood. Thank you.
Anybody else? Sir Karmakar, would you like to ask something?
Yes. So it was really inspiring. In fact, IIT Bhubaneswar has entered into an MOU with AIMS Bhubaneswar to do some collaborative work on medical science and technology. So from that point of view, I was very intrigued by whatever you presented. And maybe sometime later, I will invite you to speak to a set of doctors and engineers together. We are trying to do some courses together and so on. So I think your work will be a role model for them. Yeah.
So thank you very much, sir. Actually, coming from Odisha, I always try to give back to my state. So already I am in touch with some professor from IIT Bhubaneswar as well as AIMS. I know many of them. So even I'm pushing to set up something, some kind of collaborative work. And I'm always, I told them that whenever you need me, I'm always here. And most probably, sir, actually, we are going to get a very big project from Ministry of MIT on perception engineering. So in that perception engineering, they also told that to bring some IIT and NIT. And in my that proposal, I have put Bhubaneswar in a very big picture. I told that Bhubaneswar would be one of the important node, because sir, actually I want to start something over there, and I'll be happy.
Very good.
To be part of this.
Anybody
Hello,
else?
sir, I have one question.
Yes, sir.
Hello, sir.
Sir.
That brain, when you are taking a CT scan or MRI picture, have you
Sir.
attempted to take a 3D print of that 3D physical model?
Sir, yes, people are working on it, but actually my group, we don't work on the 3D modeling, 3D, I mean, by printing, but people are working on it. From this MRI images, they can reconstruct the 3D model.
Okay, thank you.
So I think it was extremely absorbing lecture, and many new things actually came up. And such a young age, Professor Gandhi has done actually commendable work. So now actually I would like to request our Madam Renu Sharma to propose actually a vote of thanks.
Thank you, sir. Am I audible, sir?
Yes, perfectly.
Thank you, sir. Thank you so much, sir. Thank you, Professor Tapan Kumar Gandhi, sir, for giving a wonderful lecture, this talk. You really bridge the gap between the two fields of neuroscience and artificial intelligence. You really created a very common place where we can explore very basics of science for for help of human people over here. And I would like to say a one word that really your work is really very eye-opening, and that has been captured and being published in such a wonderful like nature science and science. And I would like to say one more thing, like we would like to collaborate with you in near future so that we can also do some basic science research, as we are also having our medical science college, and we are having dental science, and we are also working on veterinary sciences. On the onset of this program, I would like to thank Shiksha Anusandhan University, IMMT, and IIT Bhubaneswar, and also IEEE Bhubaneswar subsection for hosting this prestigious webinar on this eve. Thank you, sir, for joining us for this event. Thank you, sir. Namaste.
Thank you very much.
So, Professor Gandhi, actually, which is your native place?
Sir, from in Balasore district. राइवोनिया सुनते विराट राजा दुर्ग
राइवोनिया
हाँ राइवोनिया होगा
हाँ राइवोनिया दुर्ग
हाँ
मु भी से बॉर्डर डिस्ट्रिक्ट रो मैंने मयूरभंज रो
ओके मयूरभंज तो साइकिल में कार जाती हूं सर
अच्छा यार बहुत बहुत भलो बढ़िया बढ़िया काम होची एंड यू कीप ऑन डूइंग एंड हमरो एठी बहुत फैसिलिटी मते रहिची अवश्य न्यूरो साइड किसी लोक अच्छा पर्टिकुलरली न्यूरो सर्जरी रे तो बहुत भल अच्छा यू नो अशोक महापात्र वेल सो वाज एक्चुअली डायरेक्टर ऑफ एम्स एंड आल्सो ही वाज न्यूरो सर्जरी प्रोफेसर एक्चुअली इन
सर टोटली जे मासी की साइड टॉक देबी तो आई विल गेट अ चांस टू मीट एवरीवन ओवर देयर
ओ यू सी मोर
हां नहीं सर्कल तो ऑनलाइन दे पर ठीक है सर पर आवतर आसी केतले
ना ना यूसी माथी तो अछंती एरे माने हमरो आईआईटी दिल्ली रे थिले
हां
उमाचरण माथी कथा कोसन ना
हां नहीं सर नहीं पंडा सर जे कहले टकदा पई मु कहले मु हाउची बेटर मु आसी जेटी आ समस्त मीट करे चांस मिलियो मु आपण मीट करनी आपण मानकू
ना ना बट डेफिनेटली एक्चुअली हमरो आईएनए नेशनल कन्वेंशन होची
हां एटा
you must come
हाँ सर
okay?
हाँ yes
माने you are very much a member of our
yes
INE Bhubaneswar chapter you know very well so I think with all your help and cooperation this chapter also will grow
okay. Yes sir yes
thank you so much
Triple sir actually I am thinking to spend some time in IIT sir most probably I want to be there for 1 or 2 years so that I can set up the things and I can help there.
Yeah, have you come? Have you visited IIT Bhubaneswar anytime?
Once I visited, once.
When?
Sir, recently I just delivered the cognitive science group. They invited me once. So there I interacted with them.
So that means a few months during when I was a director here, you have visited?
Ah, sir. Yes, sir. When you were director there.
Oh. But I think we have not met, no?
Yeah, I have not met you, sir.
Ah, that is what. Next time you are coming this side, let us know.
Yes, sir.
Yeah.
Yes, sir.
Yeah.
Thank you, sir.
Sir, we will definitely like to see you at annual convention of INA in the month of December from 9th and
yeah, when I saw that it is happening Bhubaneswar, I thought, oh, yes, good to come over there.
Or you may be coming during puja vacation here also. Please do come.
Sir. Sir. Yeah, but actually puja, it's very difficult during puja. But after puja, whenever I'll come, I'll meet you.
Diwali.
Diwali, yes. Diwali time I can.
Actually, you may be knowing LM Das. LM Das is my classmate.
Okay. Yes, sir. I know him. Yeah.
Lalit Mondas.
Uh-huh.
And Professor Prem Bharat is my senior and guru.
Okay. Oh.
Okay. Thank you so much.
Thank you, sir. Thank you so much. I
Thank you.
thank
No,
all the
thank you.
guests present over there. Yeah,
Professor
thank you, sir.
Karmakar, so nice that you could make it today. And in
No,
one
I am,
of these
sir.
days, actually, I will come to you for our
Sure, sure.
INA conference.
Yes, yes.
I'll come to IIT Bhubaneswar to meet you.
Yes, yes.
Okay.
Welcome.
Okay.